In 1961, molecular biologists discovered a short-lived chemical messenger inside living cells that carried genetic instructions directly from deoxyribonucleic acid to protein-building factories known as ribosomes. This fragile single-stranded molecule was messenger ribonucleic acid, or mRNA. For decades following its discovery, mRNA remained a fleeting cellular intermediate, widely viewed by mainstream science as far too unstable and delicate to ever serve as a reliable therapeutic agent in clinical medicine. The prevailing consensus in cellular biology asserted that introducing laboratory-created genetic material into a living organism would trigger catastrophic cellular defenses. In 1989, pioneering researchers demonstrated that synthetic mRNA, wrapped inside microscopic lipid bubbles, could deliver functional genetic instructions to cultured animal cells. Yet despite this early proof of concept, the broader scientific community remained deeply skeptical, dismissing mRNA therapeutics as an evolutionary dead end that could never work in living mammals. Enter Hungarian-born biochemist Katalin Karikó, who arrived at the University of Pennsylvania in 1989 with an unshakable conviction. She proposed that synthetic mRNA could be engineered to turn human cells into localized microfactories capable of producing therapeutic proteins on demand, offering potential cures for heart disease, strokes, and genetic conditions. However, securing academic research grants for such an unorthodox concept proved nearly impossible during the early 1990s. The central engineering barrier was not merely delivering the synthetic molecule past the cell membrane, but surviving the host organism's hypervigilant defense system. Every time synthetic mRNA was injected into living tissue, the immune response recognized the external genetic sequence as an invasive viral pathogen. The resulting inflammatory flare-up was severe, destroying the synthetic strand long before cellular ribosomes could translate it into a single beneficial protein molecule. Karikó spent long, isolated years in modest benchtop laboratories, working tirelessly to refine synthetic RNA transcripts. Academic administrators repeatedly demoted her and stripped away her laboratory funding, viewing her relentless focus on synthetic mRNA as a career-ending obsession with a flawed hypothesis. Despite mounting professional pressure and constant institutional rejection, she refused to abandon the core idea that nature's information messenger could heal human disease. A chance interaction at an office photocopier in 1997 altered the entire trajectory of modern biotechnology. Karikó struck up a conversation with Weissman, an immunologist who had recently established a laboratory at the University of Pennsylvania to develop vaccines against infectious diseases. Their casual encounter revealed a profound shared curiosity regarding cellular signaling and dendritic cells, the key immune sentinels responsible for detecting foreign invaders in human tissue. Combining Karikó's deep technical expertise in synthetic mRNA chemistry with Weissman's sophisticated knowledge of cellular immunology, the researchers established an extraordinary scientific collaboration. They began systematically introducing synthetic RNA sequences into human dendritic cells to isolate the precise mechanisms triggering severe inflammatory reactions. Their early experimental trials confirmed the immense challenge, as immune cells erupted with alarm signals upon contact with the lab-made code. Yet a startling mystery emerged during their systematic comparative tests. While lab-synthesized mRNA provoked violent inflammatory responses from dendritic cells, transfer RNA extracted from natural, living human cells produced virtually no immune reaction whatsoever. Karikó and Weissman realized that natural biological systems possessed a hidden chemical signature, an evolutionary modification that shielded cellular RNA from being targeted by its own internal immune surveillance systems. The microscopic secret resided in subtle chemical alterations of the nucleoside building blocks. Naturally occurring RNA within complex animal cells contains post-transcriptional chemical modifications on its nucleoside bases, including uridine, cytidine, and adenosine. In stark contrast, standard synthetic mRNA produced in laboratory test tubes completely lacked these chemical shields, making it look violently foreign to specialized toll-like receptors patrolling the interior of human cells. Karikó and Weissman hypothesized that systematically altering synthetic mRNA at the atomic level could trick host immune receptors into accepting the foreign molecule. They focused their efforts on pseudouridine, a naturally occurring isomer, where the chemical bond between the ribosugar and the uracil base is subtly rearranged.
Replacing standard synthetic uridine with pseudouridine represented a radical attempt to rewrite the artificial genetic code while maintaining its biological translation capacity. The laboratory results proved monumental. When Karikó and Weissman introduced the pseudouridine-modified synthetic mRNA into human dendritic cells, the inflammatory alarm system remained completely silent. Even more astonishingly, cellular ribosomes recognized the modified genetic transcript and translated it into functional target proteins at rates significantly higher than unmodified RNA. They had successfully passed through the host defense network without provoking destruction. This fundamental discovery resolved both primary hurdles of mRNA medicine in a single stroke. By circumventing toll-like receptor detection, the modified nucleoside architecture prevented destructive inflammatory cascades while simultaneously expanding the molecule's protein expression yield. Published in 2005, this seminal breakthrough effectively dismantled the decades-old scientific dogma that had restricted synthetic mRNA from being utilized as a viable therapeutic drug platform. Despite the clear theoretical triumph documented in their 2005 research paper, mainstream pharmaceutical companies were surprisingly slow to grasp its revolutionary potential. Patent filings faced skepticism, funding remained tightly constrained, and established biotechnology firms continued pouring billions of dollars into conventional viral vector gene therapies. Nevertheless, the intellectual spark struck at the University of Pennsylvania quietly catalyzed small, agile biotechnology startups across Europe and North America. Innovative researchers quickly recognized that pairing pseudouridine-modified mRNA with advanced lipid nanoparticles solved the remaining delivery challenge. These microscopic fatty envelopes protected the fragile modified transcripts in the bloodstream, enabling targeted cell entry without degradation. The rudimentary benchtop chemistry pioneered during the 1990s was rapidly maturing into a universal, highly modular platform capable of encoding virtually any therapeutic protein or viral antigen on demand. This conceptual evolution fundamentally altered how scientists approached drug discovery. Traditional pharmaceutical manufacturing required growing complex biological proteins inside massive bioreactors, a process taking months or years to standardize. In contrast, synthetic mRNA enabled the patient's own body to act as the bioreactor. Scientists could design, synthesize, and test pure genetic software in a matter of days using simple chemical assembly techniques. When a global public health crisis emerged in early 2020, this 30-year saga of persistence reached its defining historical test. Within 48 hours of the novel coronavirus genome sequence being published online, researchers utilized Karikó and Weissman's modified pseudouridine platform to design fully synthetic vaccine candidates. Decades of unheralded basic research were suddenly mobilized to construct life-saving interventions at unprecedented global speed. The resulting clinical trials demonstrated unprecedented efficacy, leading to the rapid deployment of billions of doses worldwide. Millions of lives were saved precisely because two determined scientists in the 1990s had refused to accept the prevailing consensus that synthetic mRNA was an unusable molecule. The long, lonely years spent defending their work against institutional skepticism culminated in one of the greatest medical achievements in human history. Today, the legacy of that 1990s breakthrough extends far beyond infectious disease management. Medical researchers are currently employing modified mRNA technology to formulate personalized cancer vaccines tailored to specific patient tumors, repair heart tissue damaged by myocardial infarction, and restore missing enzymes in rare genetic disorders. A single core platform is unlocking entirely new frontiers in regenerative and molecular medicine. The story of synthetic mRNA proves that technological revolutions rarely arrive as sudden, isolated flashes of genius. Instead, they are forged through relentless grit, intellectual open-mindedness, and the courage to challenge established scientific dogma. What began as a neglected theoretical concept in a modest laboratory at the University of Pennsylvania has forever transformed humanity's capacity to program cellular biology and defeat human disease.